muscle cell, so that's why we call it the sarcoplasmic reticulum. And then it turns out that your sarcoplasmic reticulum is normally really high in calcium concentration. In terms of calcium concentration, it's because it constantly has these calcium pumps that pumps calcium into the sarcoplasmic reticulum. As a result, these calcium ions would normally want to leave the sarcoplasmic reticulum due to them wanting to balance out the equilibrium. So now, as the action potential travels through the T-tubules really close to the sarcoplasmic reticulum, on the sarcoplasmic reticulum you have this calcium channel here called ryanodine. And then normally in skeletal muscle you have another guy here called DHP. He is a, or he, she, doesn't matter, is a voltage sensitive receptor, and ryanodine before it's a calcium channel. Hopefully you guys can see this a little better now. But yeah, ryanodine is a calcium channel, and DHP is a voltage-sensitive receptor. So when the T-tubule is carrying the action potential, that's what these arrows represent, when they travel near the sarcoplasmic reticulum, the voltage-sensitive DHP, once the voltage travels near it, it moves off of the ryanodine receptor. It moves off of the ryanodine calcium channel. So now that that's gone, your calcium can now travel out of the sarcoplasmic reticulum and go into the sarcoplasm, which is basically just the cytoplasm of a muscle cell. So now that you have calcium ions all in the sarcoplasm, what else? Something else you have in your sarcoplasm is your sarcomere, which has your actin and your myosin, and now you'll see how calcium plays a role in muscle contraction. So now we're gonna go in depth for this right chunk here. Right here. So this is in the sarcoplasm. Both of these, in the sarcoplasm. Just going to go in depth for this part here. So, you have your actin, just two strands. It kind of looks like DNA here because I draw it as a double helix, but it's actually not. These are actually beads if you remember from your textbook, the blue-looking beads. And then these actin, if you remember, they have actin binding sites. Not actin binding sites, myosin binding sites. So I'm going to use X's to represent the binding sites. Hope you guys can see it. So this is actin, myosin binding sites. And if you remember, on your actin you have a rope-like protein called tropomyosin that wraps around it. So I know in this figure it's not that great of a job. I'm not doing that great of a job drawing it, but it basically covers up your myosin binding sites. So your myosin down here, myosin heads, they basically want to reach up and grab these binding sites. But because you have tropomyosin here, tropomyosin, the head can't grab onto the binding sites. And then also, on top of the actin and tropomyosin, you have this protein called troponin. So now, relating back to here in the muscle cell, you remember how the calcium went into the sarcoplasm? Well, it turns out that these calcium, they travel to the troponin, and then, oh yeah, I missed this point, but troponin has 3 binding sites, one to actin, one to tropomyosin, and one for calcium. So now calcium comes along and it comes along from here, dissolved in the sarcoplasm. It binds to troponin. As a result of calcium binding to troponin, you have a conformational change in troponin. And as a result of troponin being binded to tropomyosin will move with troponin, and then your myosin binding sites will be exposed. So it's going to look like this. This is your actin again. This is a terrible drawing, but bear with me. So yeah, your myosin binding sites are now going to be exposed. Right, and then to better explain how, actually let me draw tropomyosin first. So now your tropomyosin, instead of blocking the sites, it's going to be chilling right here. Not blocking the binding sites. Tropomyosin. And it's still going to have the troponin binded to it. And each troponin is going to have the calcium ions bound to them. So yeah, to better explain the conformational change that occurs that allows the myosin binding sites to be exposed is here, 2 pens. Kind of think that, well, imagine that this green pen here is troponin, and then this purple pen here is tropomyosin. When calcium binds to troponin, it causes a conformational change. So because tropomyosin is bound to troponin, when troponin makes a conformational change, tropomyosin is going to move with it. And now whichever myosin binding sites were located here before are now revealed for the myosin to grab on. So then, as a result, the myosin heads can grab on. I'm just going to draw a couple here just to save time, but it turns out that when they grab on at this point, originally before they had a molecule binding to them, and it turns out that it was the hydrolyzed form of ATP. So in your book they represent it as ADP with a bond and phosphate. So yeah, that's hydrolyzed form of ATP. Okay, let's see. And then, so now your myosin heads, they're bound to the myosin binding sites, the X's here, and now you want to contract. So now you have to ask yourself, where does the energy come from to cause the contraction? And the answer is right here, in the ADP phosphate bond right here. So it turns out that your myosin heads have ATPase activity. So what they can do is they can actually break the bond right here. So they break the bond. So whatever energy that's released, so energy is released, this energy goes to the myosin heads. They each have their own ADP molecule, but yeah, the energy goes to the myosin heads, giving them the energy to pull the actin in. So this would be right here, giving them the energy to pull the actin in towards the M band, causing the Z lines to come closer. Causing the Z bands to come closer, shrinking the I band because your actin's starting to overlap more, shrinking the H zone, and then causing your sarcomere to basically contract, and you get a muscle contraction. So yeah, your actin moves in, you have contraction. Let's see. At this point here, you have your actin. Your myosin heads are bound to it. And remember, we broke the bond here, so now you just have ADP bound to your myosin heads. Myosin, ADP. So now you're done with contraction, you want to let go. So now what you need to do is you need to replace each ADP molecule with the ATP molecule. So ATP goes on and ADP falls off. And ADP falls off. Next step, as a result of this, that's my crappy actin that's just turning into squigglies. Your myosin head. Let's go. But they're not in the relaxed position like in this figure here, which is drawn really crappy. So this is the relaxed position. As you see how here they're still bent up. So in order to bend them back down to the relaxed position, right now they're bound with ATP. Remember how I said that the myosin heads have ATP ACE activity? Well, the myosin heads hydrolyze. So they hydrolyze ATP that's bound to them, allowing the heads to go back to the relaxed state. So like this. So yeah, in the relaxed state. And at this point here, you go back to the hydrolyzed form of ATP. So it's that weird ADP phosphate form that we had in the beginning of contraction. Before we broke the bond. So that's the hydrolyzed form of ATP. Okay. All right. So now the problem is the actin binding sites, the myosin binding sites are still exposed. So in order to fix that problem, we want to move our tropomyosin back on top to cover them up. So how we do that is we remove the calcium. Once we remove the calcium, the troponin will make a conformational change again because it's bound to tropomyosin as well and the actin. When the troponin moves, the tropomyosin will cover up the myosin binding sites. So now when calcium goes off, at the end of contraction, when you're relaxed, you have your binding sites here, 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 here. I know it's really ugly, but the idea is that your tropomyosin is now wrapped around your troponin in the correct way so that the binding sites are completely blocked now. So they're completely blocked, so then the heads can't bind to it. And you still have your troponin here, but the calciums go back into the sarcoplasmic reticulum by the calcium pumps here. Calcium pumps. Yeah. Okay. I hope that helped you guys. But so yeah, try to think of the sarcomere, the synapse figure, and the muscle cell figure, and the more in-depth sarcomere figure all together, so you could kind of have a better idea of what's going on, and you can link it all together. Okay? Alright, I wish you guys good luck on your last bio exam. It's been a pleasure tutoring you guys this semester.